Okay, so the first thing I want to do is inoculate my E. coli plate. So I'm going to start the Bunsen burner, turn on the gas till I barely hear it, and then just get that going. So we'll adjust it so it's a nice cone. This is my E. coli. I purchased this from a biological supply company. So what I want to do then is to take this E. coli, inoculate it on a plate, grow the plate for 24 hours, and then it'll be ready to actually do the procedures for the transformation. So this is a biological loop. So you can see at the end there it's a loop. I'm going to straighten this wire. So the straighter it is, the better. Then we go in and we're going to heat the loop up. And we do have to be careful of heating the loop too hot, because when I get my sample of E. coli, if it's too hot, then that's problematic. So now I pick this up. I don't want to put the lid down. Okay. I'm then going to flame the lid of my E. coli. I'm going to put my loop in gently and just scratch that on the surface. Flame my tip again, put my lid back on so that we're all set to go, and set that down. Now what I want to do is take my plate. You can see here's my plate. I've labeled it E. coli, so it's ready to go. I basically just want to take my plate and I'll open this up so you can see it. I'm just going to gently rub the loop across the edge of the plate. Okay. Now I'm going to put the lid back on. I'll flame the loop. Now bacteria, there's tons of bacteria. We're trying to get isolated colonies. So I basically want to spread those bacteria out. So I'm going to go here and I'll cool down my loop on the auger. Then I'm just going to take this one spread through here and go in a nice big S fashion through there. We'll turn the plate, flame the loop again, cool the loop down, and then just basically take this loop through where I just took it through and spread it out over the whole plate in a nice big S fashion. So over here then, where I finished that spreading, I should have isolated bacteria, so I should have nice isolated colonies. Flame my loop, and I am done. Now I'm going to take my plate over to the incubator, incubate it for 24 hours. So here's our plate after 24 hours of incubation. You can see where we originally streaked right here, and you can see that all that bacteria, we then took the loop and streaked up here in a continuous fashion. And you can see how as the bacteria get fewer and fewer in numbers, then we get these little teeny tiny dots called colonies. A single colony would be like right there. That single colony could have from 10 to 100 million bacteria in that colony. Okay. Now in your lab report, you need to predict what color the bacteria will be. So in predicting that color, then what you want to do is note the starting color. So this starting color is pretty much whitish in color. So it may be a little off-white to white. So you want to record that off-white to white color for these bacteria. So now I've taken the plate and placed it on a nice little UV light table. So underneath this plate is an ultraviolet light. We're going to use the ultraviolet light to see if they glow, because after the transformation, the plasmid we use will cause the bacteria to glow if conditions are in a certain way. So I'm going to then zoom in on this plate here so we can see this guy. Okay, the UV light is not on right now, so it should look like it did before, where we have our colonies. We can see that they are whitish and we can't see any glowing. Now the UV light, though, needs a lid, and so we drop this lid because UV light can be damaging to your eyes, therefore it will never turn on unless the lid is on. So I drop the lid, I turn it on, we can see the background. It'll be nicer to see if we turn off the lights, so let me turn off the lights here. Alright, and so you can see clearly that we don't have any glowing going on with those cells. Okay. Now in your lab report, then under procedure 1, you're going to want to indicate what you expect to happen in 3 types of bacteria. One is being untransformed, which is these bacteria right here are untransformed. What we're starting with, we're not transforming them, therefore we're not putting in any plasmid into the bacterial cells. Then we have 2 plasmids. We have plasmid P and we have plasmid A. So in the very first part, procedure 1 planning, you're going to predict what's going to happen on plates with what's called ampicillin, where ampicillin then is an antimicrobic that's going to kill the bacteria if they do not have the plasmid with ampicillin resistance. Then we have E. coli transformed with plasmid P and E. coli transformed with plasmid A. The only difference between plasmid P and plasmid A is the restriction enzyme that we used to try to ligate in the segment of DNA that is going to be used to degrade estrogens and make superbacteria that are going to save the world, degrade estrogens and save all of the fish so that they don't get feminized. Okay, so this is the diagram of the plasmid on page 103. Our goal in this case is to get this plasmid into bacteria. Putting the plasmid into bacteria is called transformation. And then a untransformed bacterium does not have the plasmid. A transformed bacterium does have the plasmid. So a couple things to pay attention to on this plasmid. Here's a gene. This gene then has a segment that's going to make a green protein in the bacteria and make the bacteria green, and a segment that's going to make the bacteria glow. Okay. Over here we have a gene. It's called an ampicillin resistance gene. Ampicillin is an antimicrobic that kills bacteria. Okay. If this gene is present in the bacteria, then this promoter causes this gene to run, and this gene therefore is going to cause the bacteria with it to be ampicillin resistant. They can degrade ampicillin outside their cells so that the ampicillin will not kill them. Down here, this gene has an important region called a promoter. The promoter needs to be intact in order for this gene and this gene to express itself, just like this promoter has to be intact for this gene to express itself. This promoter is different than this promoter, so if this promoter gets disrupted, then this gene and this gene will not express itself, but this gene will express itself even if this promoter is disrupted, right? The only way to disrupt this gene is to disrupt that promoter. Now what we're going to do is we have 2 different versions of this plasmid. One version is called plasmid A, and the A refers to the APA1 right here. And we have plasmid P, and the P refers to this PST1 right here. Okay. So APA1 is a restriction enzyme, PST1 is a restriction enzyme. On the previous page in the lab manual, if you're reading, then it talks about how restriction enzymes are used to basically cut DNA. So if we use APA1, it recognizes the sequence right there. It will cut the DNA and we'll be able to splice in a segment of DNA right there in this promoter region. If we're successful in ligating that segment of DNA, this promoter gets cut. This and this do not work. Okay. Now, restriction enzymes are interesting because right here we can see, if we look up here, we can see EcoR1. Restriction enzymes come from bacteria. And the reason that bacteria have restriction enzymes is mainly because if there's a bacteriophage or a virus that attacks the bacteria, these restriction enzymes then cut the DNA of the virus and so that they're protected from the virus. So we as biologists have gone in, grab these restriction enzymes from bacteria, like ECO, it stands for E. coli. R is restriction enzyme number 1. So this is the first restriction enzyme from E. coli. It would cut this plasmid right there. APA1 is from a different species of bacteria. PST1 is from a different species, where the A is for the genus and the PST is for the species. So here then what we'll do is we grab this enzyme, we throw the enzyme in with the plasmid and the ligated segment of DNA. The ligated segment of DNA sticks in like a Lego. Okay, so enzyme A we tried to ligate it with APA1. Enzyme P, we tried to ligate it with PST1. If these were ligated, this promoter will get disrupted. This promoter will not get disrupted. In the table that you have to fill out with your predictions then, right? A plate that is plus for ampicillin means that ampicillin is on the plate. Okay, normal bacteria cannot resist ampicillin, but if transformed bacteria have the ampicillin, then they will be ampicillin resistant. Okay, that should give you enough information to fill out the table.